Next, we'll be hearing from Crosse. He's a postdoctoral fellow here at Einstein who's doing some work in autism, looking at brain function in response to speech.
Okay, thanks for the introduction, Sophie, and thanks to the organisers for inviting me to speak here. So I'm going to move now from talking about speech production to speech perception and some of the new methods that we've been using in our lab to study speech and language processing in children and in children with autism. So it goes without saying, the ability to speak and to also perceive speech is one of the defining characteristics of the human brain. And without this ability, we'd find it very difficult to function within our society. But of course, as Helen mentioned, this ability is impaired in individuals with autism, but also the ability to perceive speech. And so it's thought that this impairment in speech perception may also contribute to some of the more well-known phenotypes, such as impairments in communications and social deficits. So normally when we're studying speech processing using non-invasive EEG recordings, what we'll do is we'll time lock the EEG response to some discrete event. So in the context of speech, we might use a syllable or a word. And while this is a really good way of extracting a neural response to the speech token, we're not really using speech in an ecological way, because we know for a fact that natural speech engages a really vast network of brain areas, and so it's unlikely that we're engaging them using these types of stimuli. And so what we're trying to do is use stimuli that are more ecologically valid. Another thing that we don't get from these types of stimuli, of course, is the ability to make predictions. So when we're listening to speech, we're constantly updating the system and making predictions about what we're going to hear. And there's a lot of evidence to show that the top-down prior knowledge influences speech processing even at a very basic sensory level. And we know that individuals with autism also make less accurate predictions about their sensory environment. And so these are aspects of speech that we want to preserve in the research environment. And so the only way we can really do this is to use natural speech. But of course, one of the issues then is that the actual EEG signal is a lot more complicated in terms of how we interpret it. So one of the approaches that we've been using, and it's really growing in popularity now in cognitive neuroscience, is to use these encoding or decoding modeling frameworks. And so what this consists of is your data collection phase as usual, where we record our EEG response to some ongoing speech stimulus. So this could be a natural movie or even an audio book. And again, this is something as well, it's not only ecologically valid, but really when studying this in clinical populations, it's important because it's more engaging. And so then we'll extract some features from both our EEG signals and our stimuli. And this is a very important stage, particularly in the context of the stimulus, because we have to really define what it is we think is being encoded in the neural signature. And this is something I'll talk a little bit more about in a minute. So then what we can do is we can define some sort of mathematical mapping or relationship between our stimulus feature and our neural signal using some regularized regression technique. And so we then have this model that essentially is much more versatile than the response that we would get to discrete stimuli, because we can use it to do several things. So for instance, we can use it to actually reconstruct an estimate of what the stimulus features would look like in a new unseen trial of EEG. And we can also use it in the reverse direction to show us how that information is actually encoded in the cortical activity. And so I spent most of my PhD kind of developing these open source tools for mapping speech to neural signals. But what I'm actually going to focus more on now is how we can extract different types of features from the speech stimulus in order to probe speech processing in a kind of hierarchical manner. And so what I mean when I talk about a hierarchical manner is we know that speech has this hierarchical structure. It contains different segmental units such as phrases, words, syllables, phonemes, and so forth. And the system within the brain that processes speech is also organized in a hierarchical way, just as the visual system is. And so, you can imagine the sound as it initially goes in is processed in terms of its acoustics, but then those acoustics are at some point mapped onto a phonetic representation, and then a lexical and semantic representation. So this is a kind of oversimplification of what this system might look like. And something I'm also interested in is how visual speech is processed in this way and how these two signals interact within the brain. And so really what we want to do is we want to study speech processing at discrete stages of the speech processing hierarchy in order to really understand where along the auditory cortical pathway or visual cortical pathway is speech perception breaking down in clinical groups such as autism. So I'm going to now look at some of the data that we've collected from an ongoing study. So this is some very preliminary data, and really it's just a qualitative look at some of these data and really how we can apply this framework to studying speech perception in autism. And so what we did was we presented children's stories to both children and teenagers with and without a diagnosis of autism. And so they're recited essentially by a trained actress. The children listen to them in an EEG booth while we record 64-channel EEG. We record eye tracking and also get them to detect target words to make sure they engage with the task. And so we have several different stimulus conditions. We have an audio-only condition where they can hear the story, but they only see a still image of the speaker. Then we have a visual-only condition where they can see a video of the speaker, but they can't hear her. And then an audio-visual condition where they can both see and hear her. And so we also have different levels of background noise in order to modulate the amount of the intelligibility of the signal. And so this is what the speech signal typically looks like with no noise. And then if we add different levels of noise, we can see how the signal begins to be drowned out, and at minus 15 decibels it's very unintelligible, so it's very hard to actually hear what the speaker is saying. So the next step then offline is to go and extract meaningful features from the stimulus that we think will actually represent speech processing at these discrete stages of the hierarchy. And so if we're to start at the most basic acoustic level, we might just look at something like the speech envelope. So the red line here represents just the low frequency fluctuations in power of the signal. We can go a step further then and include some frequency information where we look at the fluctuations in energy at different frequency bands. And then if we want to move on to something that's more speech specific, we can derive phonetic representations using available phoneme alignment software. So really what we have here is all the phonemes in the English language as a function of time. And so each little fleck of yellow just represents where each of those phonemes occurred in our stimulus. And we can map this binary representation to our EEG signal just as you would with the envelope or the spectrogram. So I'm just going to quickly show some data. So this is the response function for the envelope representation. And so for those of you familiar with auditory evoked potentials, it's quite similar to what you would see in a child or teenager for the audiovisual condition. Two prominent positive components that scale with the amount of background noise, and it's a little diminished in the audio-only condition because it's less intelligible. So this is from 12 typically developing subjects. And you can see the topographies are also typical of what you would get with an auditory evoked response. And so it just demonstrates that we can analyze these response functions in the same manner as you would an ERP. And so we have some very limited data from autistic participants. I don't want to read into it too much because it's early days to really comment on what's going on, but the response from the data we've collected doesn't appear to be as reliable. And although you might think, right, the response for an ERP is normally just as reliable in an autistic sample, this isn't the equivalent of an ERP. We're looking at the response to essentially how they track ongoing speech. So it's a very different process that we're trying to capture here. But anyway, I'll move on without saying too much about that. So if we were to look then at a spectrotemporal representation, we can derive a temporal response function for each frequency band to see how does fluctuations at different frequencies map onto the cortical response. And so here you've got frequency by time, and the color represents the amplitude or the power. So you can kind of see there appears to be 3 kind of distinct frequency bands where the response is strongest, and these probably relate to the formant frequencies of the speaker's voice. And so if we look at these response functions for audiovisual speech with no noise in our neurotypical and autistic group, we see a slightly different story here. We actually see quite a reliable response in the autistic group, but only at a lower frequency. And so that might give us a more detailed picture of what could be going on. They mightn't encode information at higher frequencies as well, although again, I don't want to read into it too much, but just to really explain how this framework can help you kind of get a more detailed picture of what's going on in the brain. If we then include the other signal to noise ratios, you can see how the response function diminishes as you increase the background noise, as the red starts to fade, and obviously in the autistic group as well. And so then we can also derive this phonetic representation. So here what we have is a temporal response function for each of the phonemes in the English language, and we've grouped them then by vowels, diphthongs, semivowels, and consonants. And so looking at this then in our 2 groups, we see even larger differences now, and this is something that we did predict, that the differences between these groups could become more pronounced the further up the system you go. And so obviously this diminishes too with signal to noise ratio because it's becoming less and less intelligible. So I'm going to move on from those data and just talk quickly finally about semantic level processing, and this is something that's quite recent. Defining a semantic representation of speech is a little more arbitrary because there's a lot of ways to do it, and one way that it's been done in a study that I was involved in a while back looked at measuring the similarity of words within the context of a sentence. And so this was done using, again, openly available open source software. And we then use this representation, which we convert to a dissimilarity index. So how dissimilar is every word to every other word in the sentence? We use this representation then in the same way, mapping it to our EEG signal and deriving a response function. And so these data here were recorded in healthy adults while they listened to natural speech. And what we found was that we kept seeing this very prominent negative component at about, in a central parietal position, at about 400 milliseconds. So for those of you familiar with ERPs and the N400 component, it seems to have a very similar topography and temporal dynamics to that component, which would make sense because that component is thought to index how incongruent a word is within the context of a sentence. And so if we play the same speech signal just reversed at the same subjects, we see that component go away because we've stripped the speech of its semantic content. And this is very reliable at a single subject level. You can see here for these 4 subjects. And then the time course you can see is very similar to what you would expect from an N400, where you get this prominent negativity at 400 milliseconds for forward speech, and then it goes away in reverse speech. And so this kind of represents the final stage of this framework that we want to apply to these children in order to understand how they perceive speech. And also this would be a very useful tool, particularly for looking at speech perception in those that are non-verbal or minimally verbal, in order to understand, are they perceiving the actual semantic content of the speech that they are hearing. So I know I've run out of time, so I'm going to skip the summary and just acknowledge everybody in my lab and everybody who contributed to the study, and thanks for your time.
I was just wondering whether you are actually measuring behaviorally in any way, whether they're understanding what they're hearing?
Yeah, so in as well as using this kind of target detection task to help them engage, we're also recording the response times to specific words, so we can see, are they actually detecting these words and what are their reaction times. The behavioral data is still to be analysed, but it's something that we are going to obviously marry to the EEG data, and we also have a lot of cognitive testing as well that each participant goes through.